My name is Carlo Maley. I'm a professor at the Arizona State University. Arizona State University is the largest university in America. We have around 180,000 students. But I went there because it's also the most interdisciplinary university in America, perhaps the world. They've largely abolished departments and mixed people together to work on different problems that cross disciplines. So it's a lot of fun intellectually to be there. I'm the director of the Arizona Cancer Evolution Center, which is actually a collaboration of 11 different institutions around the world, where we're trying to use ideas from evolution and ecology to do better cancer research, but also develop better ways of managing cancer.
So you're looking at cancer from the evolutionary point of view
Yes.
instead of.
Well, we sometimes treat cancer as if it's a singular problem that we can solve with a drug. But cancer is actually a whole population of cells in our bodies, billions of cells in our body with millions of mutations. And so it's a lot more like herding cats than it is treating a traditional disease. And so we're trying to address that, how evolvable it is, how much it changes when we apply any kind of therapy and develop better ways of managing it.
And each kind of cell is in a way behaving differently. So it's a totally different approach.
Yes, within a cancer, we think of cancer as a thing, but it's actually billions of cells with lots of different mutations. So they respond very differently when we apply a therapy. And we kill many of them, but we leave behind the cells that have mutations that make them resistant to our therapies. And that's been the main problem in cancer therapies. We actually end up selecting for the cells that are resistant to our therapy, and the tumor comes back, but now the drug doesn't work anymore, and we run out of drugs.
The tumor is coming back stronger than before, adapted.
It's adapted because the cells that could survive the treatments are the ones that generate the new cancer. So it's not the same cancer that you initially treated. And that's a mistake we often make. We think of it as the cancer coming back, but it's actually a very different cancer that has evolved in response to our therapies.
But you gave an example in your presentation. So you said that tumors are not, people are still dying
Yes.
because of cancer.
Yes.
Treatments are effective in some other, let's say, diseases like AIDS, HIVs.
Yes.
So why is that? Is this only because the cancer cell is so, how to say, smart and adaptive?
Kind of. So it is an interesting question. Why does it work in HIV to combine a bunch of drugs? But when we do that in cancer, it only buys us a few months. It doesn't keep it under control like it does
Doesn't
HIV.
cure, actually.
Yeah, so it's a problem I sometimes give to my students. And I think the answer is there's two aspects of the answer. One is that HIV is a very different organism than a human cell. And so you can develop drugs that attack the HIV genes that are not in human cells. But a cancer is human cells. So it's hard to kill a cancer cell without killing our normal cells. That's one problem. The other problem is HIV is a very small organism. It only has a few genes. It has something like 14 genes. I don't remember exactly how many. But a cancer cell has all the genes in a human, around 20,000 genes. So it has this massive toolbox of things it can tweak and change and bring online and turn off other things. It's a lot more flexible. It has a lot more to work with than a HIV virus does.
Speaking about viruses, it was also today another presentation was looking at SARS-CoV-2, SARS-CoV.
Yeah, SARS-CoV-2.
That caused COVID
Right.
Pandemic. So, is there anything you can learn from these kind of viruses and apply to the cancer cells studies?
So that was the work of James DeGregori, and what they showed was that the kinds of inflammation caused by an infection in the lung, like from the COVID virus, SARS-CoV-2, can trigger the growth of cancer cells that are hiding in the lung and are dormant. And there's a general problem of, they also showed it with flu, it wasn't just SARS-CoV-2, that these infections in the lung, because they change the environment in the lung, they can wake up the cells that are just, those cancer cells that are just dormant there and not dividing. And then you get a metastases in the lung that can become lethal. So the more general lesson is that the inflammatory environment can dramatically change what happens in the cancer cells. And so, yes, we can be learned from that. And the interesting approach then would be, how do we prevent that kind of inflammation from waking up the cells? Or how can we stop the cells from dividing and keep it dormant so that you can live the rest of your life?
Inflammation appears everywhere cancer appears, more
Yes,
or less.
yeah, it's complicated. Some forms of inflammation will clear cancer cells, and they'll suppress cancer.
Mhm.
Some forms of inflammation, particularly if it's chronic inflammation, will tend to generate more cancers. So it's been complicated to figure out over the long term, and sort of what's the good and what's the bad form of inflammation.
So anyway, speaking about cancer, this is a, how to say, an evolving disease.
Yes,
Yes, it evolves, evolves, evolves.
yes.
And when you say evolution, you think about something good,
Mm,
which in this case is not the case.
right, right.
So then, how do you fight if it's constantly evolving?
Yeah, so has been why it's so hard to cure cancer, because it's constantly evolving. But we haven't really been using ideas from evolution to deal with this evolutionary problem. So most of my career has been taking the tools and ideas from evolution and ecology that we've developed over 100 years of research and translating them into tools that we can use in cancer to do better cancer prevention and cancer therapy.
Okay, and where are you now?
Oh, we're there's lots of different projects. So we are using tools from ecology where they measure the distribution of different species across a landscape and applying that to the different cell types within the tumors. And then you can use these spatial statistics to both understand the ecology of cancer cells and their microenvironment and how they're interacting with the immune cells or the fibroblasts, but also use those measures to do clinical predictions of which tumors are going to be lethal or which pre-cancers are likely to become cancers. So we're using ecology for that. We're using evolution to think about how can we, um, what kind of pressures can we put on the tumors that will help it evolve in a way that will be good for us as opposed to killing us. And I think a common problem we've had in the past is we've been so focused on a cure, we haven't been thinking about alternatives to cures. And if you think about it for a minute, cure is not really the goal. The goal is to keep people alive
Yes.
and to have the good quality of life. If we could live out our lives with a cancer and but it not affect us, that would be just as good as curing. So we've been trying to cure for over 70 years, and we keep running into this evolutionary problem of it fights against us. So now we're thinking about how can we guide the evolution to be able to live with it but not die from it.
But can we guide the evolution?
We can. Yeah, sure. Everything we do changes the population of cells. So we can change what we're selecting for as opposed to just hammering it as hard as we can and then failing. Yeah,
Complicated anyway.
yeah, it is
Very, very
complicated.
complicated.
Yeah.
Okay, so this symposium turned 10 years.
Hmm.
It's a lot of, how to say, it's a it's a long period if you think a decade. On the
Hmm.
other hand, I don't know if in cancer it's a long time or not. How do you measure it?
I typically measure this in clinical trials. The only way to change how we're treating patients is to get good evidence from a clinical trial. Most clinical trials require at least 5 years to watch what happens in the patients. And it usually requires at least 5 years to raise the money and convince people to do the trial. So 10 years is actually just about one clinical trial's worth of time, essentially. So for me, that's a short amount of time, just from the simple logistics of how we can make progress. We have to test things in humans eventually and make sure that we have good evidence for them working before we change how we treat patients.
Any ways, any methods to shorten this period? I don't know.
There's some, so the traditional clinical trials just compares the standard of care versus a new experimental drug. There's a structure for clinical trials called adaptive platform trials that are based on Bayesian statistics.
Mhm.
And the idea there is you have multiple arms of the trial where you're trying out different experimental conditions as well as the standard of care. But every time a patient has an event, either they die from the disease or they are able to, the therapy keeps their cancer under control for a long period of time, you can update the probability that treatment is better or worse than the standard of care. And as soon as the what we call the posterior probability, as soon as the probability becomes statistically significantly different from the standard of care, if it's worse, you just stop that part of the arm and start a new arm. If it's better, then you can graduate that into a bigger trial with more patients. And they've shown that this is a much more efficient way of evaluating drugs. So there's a famous trial called the I-SPY2 trial in breast cancer, where they were able to evaluate 21 different experimental drugs in a short amount of time using this structure.
Oh, if you gain time, it's okay.
Yes,
Do
and
you
number of people that you have to test.
absolutely?
Yeah.
Do you how do you look at artificial intelligence? With hopeful or with, I don't know, reserves or norm?
Hopeful. So, I mean, artificial intelligence, it can be used in a variety of different ways. And one of my intuitions at the moment is that we haven't thought of all the different ways of how to use it effectively. We've got some great examples of there's an AlphaFold
Yeah,
artificial intelligence that solves how proteins fold up,
proteins.
and then
Yes,
you can use that to design drugs.
that was the
And
breakthrough.
yeah, that was a breakthrough, and I think that's going to clearly accelerate the design of new drugs for cancer. So that's great.
But for you, in what you are doing, how do you see AI helping?
I'm still figuring that out. I've asked my students that if I've given them the challenge of how can we improve what we're doing with AI.
Hmm.
Um, we were able to collect data and generate a figure for a paper in a matter of about 30 minutes, which would have taken my student, we estimated, about a week worth of time. So there's an accelerating aspect. Um, I often write papers with large groups of collaborators. We sort of have a community trying to develop a perspective on a new field. I had one of these papers that had 14 co-authors. Every author had written about their expertise, and it was 15,000 words, and the paper has to be about 3,000 words. And it was such a pain to try to edit that down that I was procrastinating, and month after month would go by, and I just wouldn't want to deal with it. Finally, I realized I can just have AI cut down the paper to a smaller size. Then I can check how it edited it against what had originally been there, see if I agree, tweak it, and it had accelerated that process and prevented me from uh, procrastinating even more.
But this is important because you have to have the capacity to check, meaning
Yes.
that you have to have the knowledge.
Yeah, so I think there's a paradox in AI at the moment that you have to be an expert already to know whether what it's suggesting is a good idea or a bad idea. And so you need to get that expertise. So if you don't have expertise and you're using AI, then you're much more vulnerable to it making mistakes and you missing those mistakes. So I think it's particularly useful when you can use it as a collaborator and you have good judgment and editorial judgment about what it's suggesting.
Yeah,
But
not instead of.
not instead of,
Not
yeah.
instead of, yeah. Okay. So buying time is okay if the quality is there
Yeah.
and the correctness.
Yeah, and you have to take responsibility for what eventually comes out of that collaboration with the AI.
Okay. So hopefully next year, if you come back to this symposium, we can speak about how you figure out and if you figure out
Yes,
AI helping you.
yes. Hopefully, we'll have some good stories.
Thank you very much.
Well, thank you. It's a pleasure.